All right, David, can you hear me?
I can, Aubrey. Hi, how are you?
I'm very well, thank you. Thanks for coming in. I should probably tell everybody, David and I rather like to tease each other. So David holds an important world record among researchers in the biology of aging, and I'm not talking about his number of Twitter followers. He holds the world record for the number of times he's given rather unconvincing excuses not to speak at one of my conferences. But this time he ran out of excuses, so here he is. And I'm sure you'll be very interested to hear what he has to say. Thanks.
Thank you, Aubrey. That may be the best.
All right, David, can you hear me?
I can, Aubrey. Hi, how are you?
I'm very well, thank you. Thanks for coming in. I should probably tell everybody, David and I rather like to tease each other. So David holds an important world record among researchers in the biology of aging, and I'm not talking about his number of Twitter followers. He holds the world record for the number of times he's given rather unconvincing excuses not to speak at one of my conferences. But this time he ran out of excuses, so here he is. And I'm sure you'll be very interested to hear what he has to say. Thanks.
Thank you, Aubrey. That may be the best introduction I've ever had. Appreciate it. Yeah, welcome back, everybody. It's been a great lineup of speakers, and I'm actually quite happy and honored to be here. So I did get 20 or so minutes. I'll try to leave at least 5 minutes for questions at the end. I think we can go till 30. Though that's still not a lot of time, so I'll get straight into it. All right. If you don't know what this is a species that's very near and dear to my heart. It's where my career, my PhD began, and this is a bromophenol blue-stained bunch of Saccharomyces cerevisiae cells, a photo that I took while I was in Lenny Guarenti's lab at MIT in the mid-1990s. And perhaps those of you who are young enough may not recall that we were working with Brian Kennedy and Nick Austriaco and Matt Kaberlein joined later, Shin and Mai. These were, it was an amazing group of people, and what we were trying to figure out was, why don't yeast cells live forever? And they, replicatively, meaning in a dividing manner, they managed to produce about 25 daughters. And to summarize 4 years of amazing work with the team, we discovered that, and this is largely started by Brian Kennedy during his PhD. Brian, of course, is now a well-known professor. Brian, who taught me how to wrangle a yeast cell, did a screen for mutants that led to longevity and stress resistance in this organism. And one of the mutations was in a gene called SIR4. Turns out we don't have SIR4, but we do have a homologue called SIR2, the gene product of which interacts with SIR4 and controls DNA repair, we later discovered, but most importantly, gene silencing. And the gene silencing was already known, actually, but linking it to aging was quite a surprise. What the heck has a gene silencer got to do with aging? And that was a really big breakthrough, though at the time we struggled to figure out really what it meant. At the time, some of you might remember, we were looking for things like antioxidant enzymes or pure DNA repair enzymes, and so silencing was very weird. So this was the beginning of the sirtuin field. We have 7 sirtuins in our bodies. I'm not going to talk about the 7 sirtuins today for lack of time, but we are writing up a paper on NAD boosters affecting health and longevity in a positive way, and we're now into our 3rd year of human clinical trials. And this is at a company called Metro Biotech, which was spun out of my lab because I don't know how to do clinical trials, or nor can I afford them. So that work has been published in a couple of papers, so that's for another talk. Today I want to talk about the extension of this work on epigenetics. What really did this SIR442 mutant that came out of Brian's screen tell us, if anything, about human aging? And so this is the initial model, that we have a landscape of the epigenome. This is originally Waddington's idea back in the 1950s. And the idea is that stem cells and primordial cells and very young cells can develop into a variety of different cell types. And we know this is how embryonic development works now, but Waddington didn't have much of a clue. But really, the epigenome now is much better understood in terms of how it's folding of DNA in 3 dimensions and 4 dimensions over time. So speaking of time, what's important for aging is that we need to understand what happens beyond development. And so this is the model in our lab that we've developed over the last 23 years. And that is that Waddington, of course, was correct. And his model can be extended out to the end of life. And we call this a variety of different names now. We call it X differentiation. We call it the erosion of the epigenetic landscape. And basically it's saying that cells, even though they have very precise cell types when we're young, we eventually lose that, and cells become X-differentiated, meaning they don't express the right genes that they should so that they can function and have a clear identity. And we are testing the hypothesis, and have been for many years, that the loss of this epigenetic landscape in youth is a major hallmark of aging that may give rise to some of the other hallmarks as well. So to test this idea, we spent 13 years and recently published this work in Cell in January. And many of you would have heard about this mouse. I'll quickly touch on it again. So the ICE mouse stands for inducible change of the epigenome. It was generated in my lab after we published a paper with Philip Oberdorfer, who was a postdoc in the lab, my lab at the time. And what we published was that just like yeast, we could see that the sirtuins in the nucleus were moving around during aging, and that they were being driven in part, perhaps largely, by having to repair broken chromosomes. In yeast cells, the rDNA is a major source of DNA breaks and genome instability. So we showed that SIR2 and SIR4 and SIR3, this complex moves to the rDNA during aging, and that was a major cause of the lifespan of yeast cells aging. And the de-repression of that movement led to sterility in yeast cells, which is a major hallmark of yeast aging. But in mammals, it's very different, of course. We are much more complicated. But we thought that perhaps DNA breaks themselves and the movement of sirtuins to those breaks might help mimic the aging process or cause the aging process. And so we tested that in this mouse. We made a system, hopefully you can see my pointer here, where we could use a drug-inducible system that I won't go into, but basically it expresses in most cells in the animal an enzyme called PPO1, which is a restriction endonuclease that cuts DNA rarely. In mammals, it's maybe 3 or 4 times per cell on average, and at any one time. And the cells can deal with it very easily. There is absolutely no evidence in these mice or cells that we induce this in. No evidence of cell death, no evidence of cells even slowing down in the cell cycle. And you cannot even see by Western blot P53 being activated. So I call it chromosome tickling. And this has been one of the misunderstandings of our paper in Cell, that we engineered this to be extremely weak so that we barely get any cutting and the cells have no issue repairing it. And we could not find any evidence of increased mutations in the mice that I'm going to show you. And so any changes, as far as we could tell, after 13 years of work, seem to be, as far as we can tell, largely, if not purely epigenetic effects. And there are major changes to the epigenome and undetectable changes to the genome, again, as far as we can tell. So we could take out cells from these mice, and we did that, and this is work by Jae-Hyun Yang primarily. And so this was the phenotype that we got in these mice. You can probably tell the one that was treated with the PPO cutting enzyme. I should mention that the enzyme is only induced for 3 weeks when the mice are 5 months old. So this is not continuous damage. The mice are fine, they don't feel it, as far as we can tell. There's no phenotype, but 10 months later, this is what we get. And I think you can see quite a dramatic phenotype in these mice. Now, this could just be a weird-looking mouse, but we did do deep analysis of these mice, as I'll show you. One analogy that I like to make is that this model of aging is similar to a compact disc, which those of us who are old enough remember that these were quite annoying because they'd get scratched and the music would skip. And DNA is digital, the epigenome is digital and very much analog, and analog systems aren't very good at preserving information. And so we think of this cause of aging, this hallmark of aging, as similar to a compact disc getting scratched. And so later I'll talk about ways to perhaps remove these scratches so that cells can read the digital information again. This is some of the data that came out in the paper looking at the epigenome of cells that we treat. So the cells, again, they don't have any DNA damage response that's overt. We can detect the breaks using H2AX, but that's really the extent of it. And I should say we can pull down the cutting with primers, but other than that, it's very benign. But what we see when we grow these out 96 hours later are major changes in the epigenetic landscape. And one of the gene clusters that is very much affected is the Hox locus, which is developmental genes. And as a theme, what we discovered many years ago, and now seems to be recapitulated in a variety of other studies, is that what seems to be very sensitive to these epigenetic changes during aging are developmental genes. So I used to think that development had nothing to do with aging and vice versa, but I've had to change my thoughts on that. And the way to read this very simply is that the red is the Cre, sorry, that's cut off, but the blue is the Cre, so blue is control, and these are hotspots for acetylation of histone H3 K27 acetylation, K56 acetylation, and K27 trimethylation. And you hopefully can see, even up the back, that the ice mice in red are different across this locus of more than 13 genes. And that landscape change due to cutting many days beforehand is also reflected in gene expression across here, and also in changes in the three-dimensional contacts of DNA to DNA. And that's shown here in high C, but also high C chip or high chip, looking at where K27 acetylation is contacting each other across large, many kilobases of DNA. And you can see that what used to be a nice fibroblast landscape, these are fibroblasts from these ice mice, has been converted into something else. Now we see that these clusters here and these enhancer regions can form new contacts here after we treat. And we think that this is perhaps what's happening in these mice. We're now trying to figure this out, but we are working feverishly trying to figure out if the DNA contacts are changing in the animal, not just in the cells. But that's our hypothesis. And this expression or re-expression of developmental genes may be one of these X differentiation processes that causes cells to be dysfunctional as we get older. Just very briefly, we looked at these mice for over a decade, trying to disprove the hypothesis. Maybe these are just messed up mice that look weird. But time and time again, with over 14 labs helping us, collaborators who I'm grateful to, we looked at all these different systems and organs in the animals and came to the conclusion, and our collaborators came to the conclusion that this is a very close mimic of mouse aging down to the molecular level. And I know Vadim and Steve are there. We had help from them to look at the DNA methylation clocks. And in a subsequent study, they were very helpful, I'll tell you about. And so, by all accounts, it looks like these mice are older epigenetically and physically and physiologically, just because of these non-mutagenic cuts. So you could say, well, maybe this is one of the ways we age. We're getting DNA breaks all the time, even if we don't go to the dentist or go in the sun or fly too much. There's a natural process of aging. Our cells get at least 1 or 2 per day, sometimes 10. And that, we think, is one of the reasons the epigenome gets messed up, because factors like the sirtuins, not just the sirtuins, but chromatin factors move to the wrong place like they did in yeast cells. So just in the second half of the talk, I want to talk about epigenetic reprogramming. I want to credit this part of the work in part to, and largely perhaps to, Wencheng Liu, the student in the lab who bravely tried to figure out how to reverse the epigenetic changes. And we were standing also on the shoulders of Shinya Yamanaka, thinking that some of his genes or all of the Yamanaka factors may be a way to reset those proteins to go back to where they came from and reset the gene expression patterns. But we kept causing cells to go down to age 0, which is not going to be helpful for us. We'd become the world's largest tumor. And Wencheng was about to give up after about 2.5 years of looking into this. He was buoyed in part by the work by Alex Ocampo and Belmonte at Salk Institute in 2016. They showed that in a premature aging mouse model, they could pulse the 4 Yamanaka factors, which we call OSKM for short. And those mice did better, they lived longer. And that was an exciting possibility. We kept coming into problems with that. We found that we kept getting cells that would be losing their identity, not regaining their identity. And the last experiment that Wencheng did, and possibly he was about to quit that project if it didn't work, he was very upset at the time. He tried the following experiment that I'll tell you about. I think I don't need to tell you about epigenetic reprogramming. Suffice to say that we did not have a lot of success with OK, S, and M, but we did do an experiment where we left out CMYC, the M, and that was a eureka moment because the fibroblasts we were testing did not ever go back to age 0. They stopped at about 80% epigenetically and functionally, and that was great. So we thought, let's go into animals and see what happens. And you can see here, the idea is not to go back to 0 ever, but to go back to a point where cells stop. And we think there's a barrier here that because we've had a very hard time trying to get beyond this barrier and go back to age 0 in the mouse, no matter what we've done with virally expressing OS and K. The system that we developed, which is now published and available, is a tetracycline, doxycycline inducible system using adenoviral vectors. We can use AAV9 for full body, which we've done, and Noah Davidson has shown that can extend the lifespan of very old mice. That's on bioarchives, hopefully publishing soon. We used this well before that to treat mice for eye injuries and aging of the eye. And so it's inducible, it's 2-vector system. The experiment with Bruce Cassandra and those on the bottom of the slide, including the 2 I mentioned, the 2 leaders in clock analysis. We worked with Bruce Cassandra to use AAV2 to infect the back of the eye. I should also mention a whole range of other collaborators that I'll get to at the end. So we've got old mice. We've got also mice that had damaged optic nerves. We can induce the virus for a while. I want to show you the results in old mice. This is the DNA methylation pattern shown as a heat map. The OSK treatment is very effective at restoring somewhat similar to young patterns. Morgan Levine as well as Vidim and Steve helped us look at the methylation age of these cells, and it was younger. What blew me away was the mRNA signaling, the gene expression patterns, and it wasn't just that some of the genes went back to the right expression. It was as though the cells knew which genes to alter and by how much. And that's shown in this graph here. This line basically tells you that the cells have a memory of which genes need to go back to a different pattern and by how much. Bruce Cassandra's lab showed that vision was largely restored in these old mice. And surprise to us, but now is, I think well accepted, and Vadim has been doing work on this too, so I want to give him credit. We believe that damage to cells, whether it's DNA damage or other insults, including sickness in vivo, can accelerate aging. And I believe Vadim was pretty clear that even pregnancy can do this. So, we know that it's hard on women, but accelerating aging is something that's quite something else. Though Vadim is showing, and others, that this is reversible. Another experiment that we did, in fact this was the first one we did, was to damage the optic nerve, and you can see that The lack of this color here is loss of nerves. This should be bright orange stained neurons in the optic nerve. But by turning on the OSK without MIC, we saw no evidence of any loss of cell identity, no tumors, even after 15 months of turning this on. This is after 6 weeks, and we can see that the nerves behave as though they're young because they're growing back to the brain. This is a mouse that's old. Excuse the feces. It's just a bit scared. And it's not having an optomotor response because it's not moving its head. But another mouse that we turn this on for 6 weeks, we now can see that it's moving its head, and it literally can see again after being rejuvenated with this OSK system. We've gone into non-human primates. This is work by, actually it's not confidential anymore. It is now public knowledge. It was announced in April at the ARVO conference. And this is work again by Bruce Cassandra and colleagues, where they damage with a laser the back of the eye of a monkey. This is a model for a disease called Nyon, which is basically a stroke at the back of the eye. People wake up in the morning and they can't see, and there's nothing you can do. We're showing in monkeys that you can actually restore vision, and using the same system, but with human genes now, they are able to restore the function of vision in monkeys. And what you can see in the green is the treated over here. This is a retinogram called PERG, and you look at the electrical activity at the back of the eye. And in the untreated damaged eye, this green is flatlined, but after treatment, this is only 4 weeks, we're going to go longer, you can see that the waves of electrical activity are restored, in part at least, in these monkeys, and they should be able to see better. Other work in the lab, we're looking at electrical activity not just in the eye, but also in mini brains. These are brain organoids, work by Xiao Tian and Jay Hyun Yang in the lab. And we're using human iPSCs that are engineered with the eye system and the OSK system, so we can drive aging forwards and backwards at will within these systems. And we can see the loss of electrical activity, inflammation with aging. We can go about 70 years of age in a matter of a week. And we've got Alzheimer's in the background of this because we've got different genotypes. And then we're actually hoping to submit a paper in the next month or so that shows that reprogramming of these organelles improves them, organoids. The other thing is that exciting is that we can now treat old mice and mice with genetic manipulations that mimic Alzheimer's. We can treat them with our system and ostensibly reverse the age of those neurons in the brain. We're targeting the hippocampus, and we have positive results there, improvements in memory, and even potentially, and this needs to be repeated, but potentially the recovery of lost memories in those animals. And so the model is this that we're working off. At the bottom, you can see we're born with great cell identity in our tissues, and we see this lost over time due to the reorganization of chromatin modifiers, which was the name of the theory back in 2009 that we published. We now refer to this whole system as the information theory of aging. What we think is we lose cellular identity over time due to damage and insult and movement of chromatin factors. But the really cool part about it is that it seems like some of this can be restored either by expressing OSK or OSKM if you're careful, and you can get some of this gene expression pattern back and function of tissues such as the eye. And hopefully in 18 months we'll be testing our first human to see if we can improve their vision. And so the summary is that we don't just need to slow down aging anymore, and we've been doing this with small molecules, as you know, for 20 years by activating sirtuins and raising NAD, but now we think we can have a bigger effect that can at least, perhaps not permanently, but go back to an earlier, more permanent youthful state. And we know now we can do this at least twice, and probably we can keep repeating this system. So what about, just to finish up, what about chemicals? Because gene therapy is challenging technologically and it's expensive. So Chris Petty and Thomas Dixon in the lab is a student and a postdoc, have been working for the last few years on finding chemicals that could reprogram in the same way OSK does. We know OSKM can be done, that's many labs' work over the years, but can you do it safely without causing tumorigenesis without losing cell identity. That's the trick. And we just published this in the journal Aging. I know Vadim has some wonderful work on this as well, and he helped us on this project, so shout out to him. What we found is a variety of chemical cocktails. We have better ones now. These were the initial ones, and you can see that there's restoration of this phenotype, which we look at, which is the ability to sequester M-cherry in the nucleus, because with aging, it spreads out into the cytoplasm, and we can reverse that with these chemical cocktails, as well as using OSK, of course. So thank you to many, many people. I've given shout outs to some of the key folks as well. Zhigang He is the guy that did and his lab did the pinching of the optic nerve, one of the original studies that led to a lot of this. And then too many people to thank, really, but I just want to be very full of gratitude to all of the people that did the work, and I get the honor of being able to speak about it today. And thanks, Aubrey, for your patience with my stubbornness and truly legitimate excuses. And I thank you for inviting me. Thanks.
All right. So, David, thank you for that. That was really exciting stuff. I've got one question for you. I'll start, with taking chairman's privilege and all that. This developmental barrier that you spoke about is very, very interesting. Do you know whether it has anything to do with the embryonic fetal transition that Mike West has been working on for a number of years, that's characterized by the turning on of scarring and so on? Is it a similar thing?
That's one of the hypotheses, yeah. So we are, standing on your shoulders there, and the developmental biologists. We have traced this effect, at least the necessity of the TET enzymes, which are, as you know, the DNA demethylases. TET1 and TET2 are necessary for reprogramming. What stops them from going further back, we're unclear. We think PRC2 may be involved. That keeps coming up in our analyses. And we're now tracking physically where are these proteins going during the reprogramming so that we can try to identify that barrier. But yeah, I think you're right that we're going to find some commonalities between embryonic development and this reversal process.
Cool. Any other questions? What we got? Who's got the roving mics? There's one question over there.
Hi, David. My name is Matt. I have a question, please. I'm just curious to know, how far are you away from actual using this OSK model in humans, and thinking about it, reflecting critically, like, how far are you away of actually rejuvenating a human? Is it the delivery methods that are lacking, or what are the actual things, barriers to success?
Thank you, Matt. Well, we co-founded a company, which we have to do to develop drugs, as far as I know, in 2017, called Life Biosciences. And they've been working on this ever since. So there's been a lot of safety studies in mice that have so far been positive towards getting into humans. And I showed you the monkey work, which is ongoing. Life Biosciences isn't waiting for more data. They've just put down, they have already put down $10 million to make the virus for the human studies. It's all laid out. The plan is to treat the first human patient in 18 months for nigh on, and possibly, depending on budget, glaucoma. And that'll be the first couple of clinical trials, most probably. So that's going. If your question is how long before we fully rejuvenate a body, and on social media, I said that this is a possibility, and I think it caused a bit of a ruckus. I think that it's not really a question of if, it's a question of when. When is it? Well, I think that gene therapy in the whole body is something that's a long way away because the delivery is hard with viruses. It's not even in a mouse. We have done AAV9 in a mouse, as I mentioned, and it seems to be safe, and the mice live longer based on this other study. But in humans, the FDA is not going to be too likely to allow that anytime soon. So this is why we're pushing forward with the chemical treatment, that we're now down to just 2 chemicals that work as well as OSK in vitro, and these could go into the clinic in a matter of years from now. So, you know, we're getting there. We're a lot closer than we used to be.
Any more questions? There we go, yep. Microphone, there we are.
Hi, David. I'm Phil. First, I want to say thank you because I started my longevity experiment by copying you. I saw videos of him on TikTok, and I thought that's a good way to start. So for the last month, I've got to thank him for taking cold showers every day and fasting and all these things. And I'm not going to ask you the question that everybody is asking me on the TikTok, which is what supplements are you taking now? Because you probably answer that all the time. But if you could answer that, that would be fantastic, what you're actually taking. I would like to ask you, how long is it till we get possibly this magic pill? Will it be in our lifetime? And what should we do as people to stay fit and healthy to hopefully see that day?
Thanks, Bill. Well, first of all, it's not magic, it's science. And the second of all, well, if these molecules turn out to work, I'm talking about in my lab, and we can find natural molecules that are already on the market or available, then that's very different from having to wait another 7 to 10 years to develop a drug. And a combination of 2 drugs is even harder. So it's going to depend on that. And I'm not sure yet where we're going in those 2 paths. But I think it's possible if we want to be optimistic. And as Aubrey knows, and he is one too, a diehard optimist, we like to think that we will find natural molecules that can mimic OSK, and we will be able to take those by choice in the coming years. So is it going to happen in your lifetime? Well, you know, unless you're 105, I think you've got a good chance.
And that's slightly more optimistic than me, which is saying something. So anyway, thank you so much, David. This is wonderful. And I should give you fair warning that I'm determined to get you here in person next year. So start working on your excuses now. And thanks very much.
Thank you, Albert. Thanks, everyone.